Today, I'll be presenting AACR poster 6635, enabling single-cell analysis of copy number variation in breast cancer. For this case study, we asked the question, can we call CNVs in clinical breast tissue samples? To do this, we purchased fresh frozen breast cancer tissue from CureLine, which was infiltrating ductal carcinoma grade 2. And we took 3 patient samples and processed it using our optimized internal nuclei prep protocol. And we'll be highlighting one sample in particular for this presentation. Additionally, we used an 8% spike-in of Raji to facilitate CNV baseline calling. And for the panel, we used our Tapestry single-cell DNA breast cancer research panel, which contains clinically relevant SNV and CNV, and was validated by George Rees-Filho at Memorial Sloan Kettering, who is our inaugural Mission Bio Solid Tumor Center of Excellence. Here we have our SNV calling and a schematic of our Tapestry pipeline output. If you'd like more information on this, please see poster 6171. On the right side, we have a heat map of the breakdown of the sample that we're highlighting, which was 88.5% patient, 8.4% Raji, and there was a 3.1% population of mixed cells. Here we have the chromosomal changes identified at an arm level. Specifically, we had a gain at 1Q, 7Q, and 8P, and we had a loss at 13Q. It's also important to note that we put at least 5 amplicons must be present in order to accurately call those CNV changes. At a gene level, we have the schematic here. So we had the CNV analysis tool was able to determine gain and loss, and we uncovered clinically significant findings. Specifically, we had a gain at FGFR2, which is amplified in about 5 to 10% of all breast cancers. And we had a loss at BRCA2 and RB1, which both are associated with poor prognosis. And to reiterate, Tapestry was able to identify CNV and SNV in clinically relevant samples. So in conclusion, there are many applications that this technology can be used for, such as clonal mosaicism and neoplastic transition, clonal evolution in primary tumors, metastatic dissemination and epithelial to mesenchymal transition, and drug resistance mechanisms. So the solution we offer you is our Mission Bio Tapestry, which can simultaneously measure SNVs, indels, and arm level copy number variations, including loss of heterozygosity at the single cell level to up to 10,000 cells, unambiguously identify variant zygosity and mutational co-occurrence, and detect rare cell populations. Thank you for your time, and if you'd like more information, please visit our Mission Bio booth, number 1910, or go to missionbio.com. Thank you.